Today we are going to be talking about ATM protein kinase, an exploration into the pathways that regulate DNA damage repair mechanisms and cancer. To start off, let's get into some background.
This is a general schematic of the many processes that ATM is involved in. In this presentation, we will focus mainly on the p53 pathway leading to apoptosis and its role in cancer.
So getting into how ATM kinase was discovered, researchers found that these ataxia-telangiectasia patients have improper checkpoints for G1 to S and G2 to M. They determined that this was because p53 was not properly induced. As you will learn later on, p53 is the gatekeeper in the cell. So when it isn't properly induced, that is not good. So then researchers thought, what happens if we mutate this gene? So they went ahead and introduced mutations into the AT gene. And they noticed a kinase domain. Wait a minute, this domain is similar to two other proteins discovered. Those two proteins were involved in DNA repair and checkpoint control. This discovery set the stage for viewing AT-mutated kinase, ATM kinase, as a DNA repair kinase that we will explore further in this presentation.
Now let's take a look at the structure of ATM determined through cryogenic electron microscopy. The ATM gene is a 12 kilobase region that encodes a 3000 amino acid protein. The human ATM dimer exists in a dynamic equilibrium between its closed and open conformations. It has a butterfly shape where the end solenoids form the wings and the fadkins, which are the C-terminal PIC conserved unit, make up the head. Let's talk about the closed conformation first. An intermolecular disulfide bond between the cysteine 2991s of the two protomers is responsible for shifting the ATM dimer into its closed state. Its breaking and formation is involved in ATM activity regulation. An important interaction between the flap and the flap BE restricts access to the ATM substrate binding site. On top of this restriction, the PIC regulator domain, or PRD, acts as a pseudo-substrate to block the active site in the closed dimer. The fadkins are arranged symmetrically, and the upper and lower interfaces of ATM interact with one another. Now, there's disorder of the PRD helices K-alpha-9D and K-alpha-9C, which leaves enough space for peptide substrate to bind at the substrate binding site, suggesting that the open conformation is ATM's active state. There is also a loss of contact between the flap and the flap BE, which releases constraints on the PRD pseudo-substrate, which is free to leave. Finally, the fadkins are arranged asymmetrically. The lower interface is only partially retained, and there is total loss of contacts in the upper interface.
So now let's look at different pathways by which ATM can be activated. The first is the presence of DNA damage. High concentrations of double-stranded DNA breaks can activate the ATM protein through a regulating complex called MRN. This induces splitting of the inactive ATM homodimer into the active monomer protein. The MRN complex is composed of three proteins: MRE11, RAD50, and NBS1. The MRE11 molecule has nucleus activity that enables the complex to cleave between DNA nucleotides. RAD50 is responsible for unwinding the DNA double helix, while NBS1 is utilized for nuclear localization and regulation of MRE11 and RAD50 activity. At the time of monomerization and activation from DNA damage, ATM autophosphorylates at serine 1981. It is suggested that this autophosphorylation disrupts the inactive ATM dimer to convert it into the active monomer that can bind phosphorylatable substrates. ATM is recruited to DNA break sites in a manner that requires ATP and double-strand break at the end of DNA. The action of ATP binding, but not hydrolysis, is required to drive a conformational change in the MRN complex. ATP specifically binds to the domain on a RAD50 protein. The actual size of the double-strand break is very important for this process. ATM activation favors long DNA strands with blunt or short overhangs. These overhangs should be less than 25 nucleotides. Longer overhangs behave more like single-stranded DNA breaks, and this type of repair is typically mediated by a different but similar protein called ATR. So let's take a quick break and talk about the conservation of the ATM protein and MRN complex across kingdoms. The action of DNA damage repair in both eukaryotes and prokaryotes can be facilitated by ATM. In eukaryotes, the ATM protein kinase and associated mRNA complex are typically responsible for DNA repair of double-stranded breaks. In prokaryotes, however, a similar version of the mRNA complex, containing only the MR portion, has been attributed to double-stranded break repair. This means that the NBS1 protein is only conserved in eukaryotes. The exact reason for this difference is still unknown, and some experiments have provided evidence to suggest that an MR complex in association with ATM is enough to activate the DNA damage response in eukaryotic cells. However, it is suggested that the NBS protein makes localization of the ATM complex to DNA more effective, therefore initiating a more accurate repair pathway in eukaryotes. So let's go back to talk about the mechanisms of activation for ATM. The second pathway is independent from mRNA and relies on oxidative changes to ATM that result from reactive oxidative species, or ROS. This pathway does not split the inactive homodimer, but rather modifies it into an active dimeric state.
ATM can also be activated by reactive oxygen species, or ROS. This activation pathway occurs independently and functions separately from mRNA and DNA double-strand breaks. ROS is predominantly produced in the mitochondria and peroxisomes due to oxidative phosphorylation and beta-oxidation of fatty acids, respectively. These ROS react with the sugar phosphate backbones and pyrimidine bases of DNA, causing DNA damage, especially in cellular compartments known to produce ROS. Menadione, a fat-soluble vitamin, is also known to generate mitochondrial ROS concomitant with a decrease of NADPH related to redox cycling, as well as promote the formation of activated ATM forms. NOX4, also known as NADPH oxidase enzymes, also acts as a ROS producer in phagocytes and other cells by transferring an electron from NADPH to generate superoxides. In response to oxidative stress, ATM is directly oxidized, forming a disulfide linked dimer which is phosphorylated on S1981 and thusly activated. Most reactive oxygen species are generated as byproducts during mitochondrial electron transport, and the sequential reduction of oxygen through the addition of electrons leads to the formation of a number of ROS, including superoxide, hydrogen peroxide, hydroxyl radical, hydroxyl ion, and nitric oxide. If there is a lack of ATM, we begin to see neurodegeneration via ROS-induced mtDNA damage in the brain. Neurons have a high rate of mitochondrial respiration to produce more energy in the brain, so they are more affected by oxidative phosphorylation-related ROS production.
ATM kinase plays a major role in p53 stabilization after DNA damage occurs. p53 is a tumor suppressor that is involved in controlling cell division and cell death and has many ties to cancer. MDM2 is a ubiquitin ligase that promotes the degradation of p53 under normal conditions. It does so by forming a stable complex through its N-terminal domains. After binding, it recruits a ubiquitin conjugating enzyme that modifies p53 lysine residues. When there is an absence of a stress signal, p53 can be degraded. On the other hand, if irradiation or physiological stresses such as chemotherapy, heat shock, or an acidic pH occur, ATM will be activated. Once ATM is activated, it will phosphorylate MDM2 near the ring domain. The phosphorylation of MDM2 prevents the polyubiquitination of p53. Without polyubiquitination, p53 cannot be flagged for degradation by the proteasome. As a result, it is stabilized and available to function as a tumor suppressor.
Another important role of ATM kinase is its ability to propagate a signal across damaged chromatin. When DNA breaks, histone H2AX is phosphorylated to form gamma H2AX. Imagine gamma H2AX as a road flare. Road flares are used during states of emergency, and having damaged DNA is an emergency that needs to be swiftly dealt with. Luckily for the gamma H2AX, 3 heroes show up in the time of distress: ATM, mRNA, and MDC1. MDC1 reads the gamma H2AX, and ATM mRNA complex phosphorylates MDC1 so that MDC1 stays at the gamma H2AX. When this happens, the road flare becomes brighter. That is, the signal gets stronger, as the GIF shows. And more attention is given to the damaged DNA, kind of like the Avengers fighting Thanos. As we have seen with the machines we have studied over the semester, we don't want continuous activity. We need to have a way of regulating the machine so that it operates only when it needs to. For ATM kinase, this method of regulation is a positive feedback loop. You see, when DNA suffers damage, it calls for the help of 3 enzymes, and through the following steps, the power of ATM is reinforced. Autophosphorylation is a method that activates ATM, but these steps make ATM even more active, that is, maintain its activity. Upon DNA damage, ATM phosphorylates an enzyme called CABL tyrosine kinase. And then that tyrosine kinase will phosphorylate TIP60 acetyltransferase to activate it. The phosphorylated TIP60 will acetylate the ATM to make it more active in repair. FOXO3A stimulates this feedback loop, but Notch1 will competitively inhibit CABL tyrosine kinase as it will bind to the C-terminal of ATM in the kinase domain.
Now, let's talk about how ATM has medical relevance. While ATM activates DNA damage response and therefore DNA damage repair, this can actually be detrimental in cancer treatments. The goal of radiation therapy is to damage cells beyond repair and trigger apoptosis, but ATM can sometimes begin DNA repair before the cells are thoroughly damaged. If an ATM inhibitor is added, DNA damage response is prevented. Next, radiation can be applied to the cells. With the inhibition of ATM, there is no DNA damage response, and the radiation severely damages the DNA. Then, the ATM inhibitor wears off, activating the molecule. This allows ATM to carry out its function, detecting the DNA damage. When severe damage is detected, ATM phosphorylates p53 and triggers apoptosis, killing the cancer cells. Some researchers are actually working on making this theory a reality. AstraZeneca recently put an ATM inhibitor, AZD1390, into a phase 1 clinical trial for cancer treatment. This drug features improvements to AZD0156, an earlier ATM inhibitor used to treat tumors. Both drugs target glioblastoma multiforme, or brain cancer. The main advantage of this new drug is that it has increased permeability through the blood-brain barrier, allowing more of the drug into the brain. In this image, a color-coded PET scan of a monkey brain is shown. The image on the left shows the permeation of the new drug, AZD1390, and the image on the right shows the permeation of the former drug. There is much more area lit up in the brain with the new drug than the brain with the old drug. This shows that the permeability is increased, letting the inhibitor prevent ATM action until after radiation is applied. The researchers found this was an effective way to kill cancer cells in animal models.
So now you might be wondering, what does the future of ATM kinase look like? And the answer to that is, well, major questions still remain about the high-resolution structure of ATM and what the important phosphorylation events are. However, technology is ever advancing, and new methods developed from the technology will help address these questions and discover even more about the function of ATM and how it can not only treat cancer, but other genomic diseases and disorders of malfunctioning ATM. Some of these methods include super resolution microscopy, high throughput sequencing, and CRISPR-Cas9. CRISPR is a very important machine in and of itself, but luckily you'll get to learn more about it in Group K's presentation.
Here are our sources. Thank you so much for watching this presentation. Now we'll take questions.